we'll finish with the paper from Monday, and then we'll go through the second paper. There was a third paper this week, but I didn't get a chance to read it, so we'll probably push it because it's a good one, so I want to include it, but we'll push it to next week. So when it comes to this Listerine paper, we left it at Listerine prevents lipid accumulation. So whenever they took out Listerine, there was this accumulation in cholesterol inside of cells, and that phenomenon was associated with a poorer efflux of cholesterol. Right? So cells were retaining cholesterol more than wild-type counterparts. They also know that Listerine, or they observed that Listerine promotes ABCA1 levels by slowing down its degradation. That's the last graph that we covered on Monday. If you guys recall, they did that cyclohexamide chase experiment in which they stopped all new production of proteins, and they followed the fate of any remaining ABCA1 there was inside of the cells. In wild-type cells, that ABCA1 was lost at a rate, but in cells that didn't have Listerine, ABCA1 was being lost at a faster rate, right? So they concluded that Listerine was preventing ABCA1 degradation. And then we also know from the literature that ABCA1 pumps out lipids, among others cholesterol, and therefore it will inhibit hyperlipidemia. So by now, we have these two observations. On the one hand, Listerine prevents hyperlipidemia. On the other hand, Listerine promotes levels of ABCA1. We put the two together, and we have the reasonable hypothesis would be that Listerine induces ABCA1, right? Which in turn prevents hyperlipidemia. Let me maintain the color coding. Prevents hyperlipidemia by pumping those lipids out of cells, right? So it's just a simple adding of these two observations into one hypothesis. So my question to you, the usual, how do we test these hypotheses? That's right. Yeah, the usual. We take out that middle gene in this case. So the idea would be so you would take out the middle gene, but just by itself. So take out ABCA1. Yeah, take out ABCA1. Yeah. What question does that answer? I'm turning this into one of those trivia games. Jeopardy. So if we take out ABCA1, what question are we answering? What experimental question are we answering? That's the key. So you're saying, does Listerine need ABCA1 to prevent hyperlipidemia? But if I take out ABCA1 and that's the only thing that I do, am I answering that question? That's right. By taking ABCA1 only and not doing anything else to the system, we're simply addressing the question of whether ABCA1 does anything to hyperlipidemia. And theoretically, we know that it does, right? So that's sort of the second leg of the hypothesis, yeah. So to address the whole model, what experiment can we do? Jessica. All right, so what you're saying is we take out, and let me what you're saying is we take out Listerine, right? So let's take out Listerine. What would that do to ABCA1? The levels would decrease, right? So in a way, we're taking out ABCA1 now. And what would you expect to see with hyperlipidemia? we're decreasing ABCA1 and you're saying that it would decrease?
Oh, no.
It would raise, right? Yeah. So we take out Listerine. That, if the model is correct, that should take out with it ABCA1 and hyperlipidemia should increase. But then you mentioned, what is it that you would do to the system?
Find some way to overexpress ABCA1.
That's right. So the experiment is we lose Listerine. Theoretically, we're losing ABCA1, but then we put it externally. And then we ask the question, what happens to hyperlipidemia? Right? You guys see it? That's not the only approach. What other approach can you think of? You inhibit ABCA1, and we go back to this idea that if you inhibit ABCA1 and you see what happens to hyperlipidemia, you're really asking questions about ABCA1. You're not asking questions about the whole model. And see if hyperlipidemia comes back or the other way around, or it goes back down. Again, we're going in circles, right? So we're still addressing the question of what does ABCA1 do to hyperlipidemia. But what we want is that some sort of coordinated manipulation such that we can address the whole pathway. So I'll give you a hint. It's the counterpart of this experiment. So in this experiment, we inhibited Listerine and we rescued, I don't know if that's the word that you used, Jessica, but we in genetic jargon, we would say we rescue ABCA1 expression exogenously from the outside, and then we ask what happens to hyperlipidemia, right? What's the counterpart of this experiment? Yeah?
You could either over-make it where it is hyperlipidemia,
Uh-huh.
have such a high quantities of...
I see. The issue with that approach is that now all of a sudden you're asking questions about some type of feedback mechanism. If you're manipulating hyperlipidemia in itself, the problem is that you're starting into the model by the final output, right? So we always want to think of experiments that we design such that we explore the pathway from the beginning, right? But it's along those lines. So in this experiment that we discussed before, we took out Listerine. What would be the counterpart of taking out Listerine? Put a lot of Listerine, capital letters, bolded. What would you expect happens to ABCA1? Goes up, right? So now there's a lot of ABCA1. What would you expect happens to hyperlipidemia? Almost none, right? So there's a very strong inhibition of hyperlipidemia to the point that very little hyperlipidemia. Okay, cool. And what do you do to that experiment? Because if putting a lot of Listerine and seeing that ABCA1 goes up and hyperlipidemia goes down, it's just correlation. Cut out the amino acid. And we come full circle. I think that's maybe what you had in mind originally, right? We take out the intermediate, but we need to increase that starting point. We need to increase that trigger originally, such that taking out the intermediate makes sense as a pathway, right? So exactly that. So we increase Listerine somehow, and then exogenously we inhibit ABCA1 expression via any of the usual suspects, right? It could be RNAi, it could be with a knockout, it could be with a bunch of approaches to inhibit ABCA1 expression. And then we ask the question, what happened to hyperlipidemia? And obviously, our control in that case would be an increase in Listerine without any manipulation of ABCA1, right? Because we want to see what happens baseline when we increase the amount of Listerine. I know that it's gelling in your minds, but this is the usual approach to any experimental strategy for testing a model of a pathway, right? You have steps A, B, C. So what do? Either you put a lot more A, but take out B, and then ask what happens to C, or you take out A, replace B, and you ask what happens to C. Yeah? Cool. So what did these people do? They actually took the first approach, the one that Jessica was suggesting before. So we know that that's the model. Without listering, you would lose ABCA1, and therefore hyperlipidemia would be unchecked. There's nothing to keep it under control now. What the researchers suggested to do was to replace ABCA1 externally and then ask what happens to hyperlipidemia. And the way that they replaced ABCA1 expression in this particular experiment is a little convoluted. Normally we would say, oh, just overexpress ABCA1, right? Like introduce cDNA for ABCA1 into the cells and let the cells express more. Or we might use, if we know enough about ABCA1's function, we could use some sort of agonist or pharmacological agonist that increases its activity. In this case, what they used is erythrodiol that apparently is a fairly, they claim, it's a fairly specific inhibitor of ABCA1 degradation. So it's almost like we're inhibiting the degrader of ABCA1. We're going to end up naturally with more ABCA1 inside of cells at the end of the day. Okay? So that's the approach they're taking. Instead of putting more ABCA1, what they're doing is they're taking out pharmacologically one of the things that inhibits ABCA1 expression. Yeah? And when they do that, so let's go through the samples here. The first one is the wild type, and there is a certain amount of cholesterol efflux, so there's a certain amount of cholesterol being pumped out. When they knock out listerine, there's going to be less ABCA1, therefore less pumping out of cholesterol, therefore more accumulation of cholesterol inside of cells, and that's what happens in the listerine knockout. But if they take those listerine knockouts and effectively increase the levels of ABCA1 inside of cells, the cholesterol efflux gets restored, which means more cholesterol is being pumped out of the cells, and therefore the levels of cholesterol inside of the cells should be reduced. Okay? Any questions, anyone? So far the results do support this model that listerine induces ABCA1 and ABCA1 inhibits hyperlipidemia. My next question to you, and this should ring a bell because we discussed this before. Let's say that now we'd like to propose that in addition to controlling the levels of ABCA1, listerine may be inhibiting hyperlipidemia through some other mechanisms, through what we would call ABCA1 independent mechanisms. It may be direct, it may be indirect, like what it does through ABCA1, but the point is, how do we know if listerine is inhibiting hyperlipidemia through some other mechanism? So how do you test for that possibility? Do you guys remember what we did? You do? Kind of? We talked about this in the context of whenever we want to see if there is two things working together and the first one is working through something else, what is it that we do? It's fine. It's fine. I'm prepared. You're making gestures, but... Take out ABCA1 completely. That's right, yeah. So I did have this slide ready just in case that we had to walk through it more carefully. So let's consider both scenarios, right? But that's exactly what they do, is they knock out ABCA1. So we have these two scenarios. On the left, everything listerine does for hyperlipidemia goes through ABCA1, right? So there's no way around ABCA1. On the right, we have the alternative hypothesis or model that in addition to controlling ABCA1, listerine may be doing something else. So if we have wild-type cells and we knock down Listerine using siRNA. Those are the black bars. In wild-type cells, when you take out Listerine in either model, you should see an increase in the amount of, in this case, lipids inside of cells, hence the oil red staining. Okay? In either model, taking out Listerine through siRNA should increase the levels of lipids inside of the cells. The difference is what happens to cells that don't have any ABCA1 in them, to the ABCA1 knockout cells. So let's first, let's consider the model on the left. So if you have cells that do not have ABCA1, go back to black, you have cells that don't have ABCA1, and your model says that Listerine does what it does through ABCA1 only. What would you expect to see when you inhibit Listerine? Is the increase in cholesterol levels less than in the controls, about the same or more?
Where?
On the left. So on the left, we have cells that don't have any ABCA1, and we inhibit Listerine. Would the amount of cholesterol accumulation inside of the cells be the same, less, or more than in cells in which there is no ABCA1, but you didn't inhibit Listerine?
What if it be the same?
Yes, it would be the same. Yes. Sorry.
It's okay.
I extended your life at the back end, don't worry. It would be the same. Why? Because if it is true that Listerine works only through ABCA1, if you took out ABCA1, now Listerine has nothing else to do. Listerine cannot even reach hyperlipidemia because you took out the only conduit that it had to connect to hyperlipidemia. So you can inhibit Listerine now. There's not going to be any further effect. There's not going to be any added effect. What about the model on the right? So same scenario. Now you have cells that don't have ABCA1, and you inhibit Listerine in them. What do you think happened to the lipid content? Is it the same as in the knockout? Is it even higher? Increase, right? So it would be higher. Why? Because now, yes, you took out ABCA1. This is a knockout, so there's no ABCA1 left. But because Listerine has some sort of ABCA1 independent way of reaching or inhibiting hyperlipidemia, when you take out ABCA1, you'll see some lipid accumulation, but then when on top of that you inhibit Listerine, you'll be activating this additional pathway, and you'll be inhibiting lipid exports and therefore inducing hyperlipidemia further. I do realize that it's a little counterintuitive because the end effect on hyperlipidemia is inhibition, that it gets a little counterintuitive because we're seeing increases when in reality things are inhibiting hyperlipidemia, so how come? But remember that we're inhibiting the sources of an inhibition. So in the end, what we're doing is inducing hyperlipidemia, right? That's exactly right. That's exactly right. So whenever you want to, we established already that ABCA1 is an intermediate of Listerine. Whenever you want to address the extent to which Listerine, in this case, depends on ABCA1, or in general, the original cause depends on an intermediate, what you need to do is fully take out that intermediate and compare it to what happens when you take out both, right? And this is the experiment they did. They knocked out ABCA1, they inhibited with siRNA against Listerine, and they saw that the inhibition was about the same as with the ABCA1 knockout only. So with that, they conclude that Listerine is inhibiting hyperlipidemia through ABCA1 and apparently nothing else. Did that make sense? Yeah? Cool. So the last few experiments, we're not going to go in detail. There is less genetics about them, and it gets a little heavy on the names and the drugs and everything else. But the rest of the figures, I list them here, have to do with the mechanism by which Listerine protects ABCA1. And it's all about its E3 ubiquitin ligase ability. And it is all about how ABCA1 normally gets degraded through a pathway that we'll describe in a second, and Listerine is there to prevent that, right? So we know, or we learn from them, that proteins that are sitting in the membrane can be degraded via endocytosis through a number of mechanisms. So one is what is called calpain-dependent degradation, right? And it goes through interactions with a protein called calpain that will induce the internalization of these membrane proteins and degradation. There are pharmacological inhibitors against this pathway, against this mechanism, and they used them, and they saw that ABCA1 degradation was not affected. The levels of ABCA1 were the same when you inhibited calpain. So it's not the calpain pathway. I meant to cross it. Then there is a pathway that is dependent on ubiquitin, but whenever you're marking, remember that they're interested in marking of proteins at the membrane for degradation because they know that Listerine is a E3 ubiquitin ligase. So it makes sense to look into the pathways that are depending on ubiquitination. So when it comes to ubiquitination or ubiquitination, there is two branches. One is the non-lysosomal, the one that doesn't depend on lysosomes, that simply marks proteins for destruction with ubiquitin attachments and then sends them to the proteasome. And that is the non-lysosomal pathway, the proteasome. There are, once again, pharmacological inhibitors of the proteasome. They're pretty common, by the way. Many people use them in labs. And when they use that pharmacological inhibitor that kills the proteasome, levels of ABCA1 stay the same, meaning ABCA1 is not regulated by the proteasome. Right? So then the only thing left is ubiquitin marking for lysosomal transport or sorting into lysosomes. And when it comes to sorting molecules from the cell membrane into lysosomes for destruction, there is, once again, two options, two possibilities. One is a pathway that depends on a protein called ATG5, and another one depends on a protein called HRS. And if you're uber curious, you can go to the paper and check these experiments for yourselves, but they actually knock out ATG5 in some cells, knock out HRS in other cells, and they see that when they knock out ATG5, nothing happens to ABCA1, but when they knock out HRS, the levels of ABCA1 increase significantly. And what that's telling them is that ABCA1 is normally destroyed through the HRS-dependent ESCOR pathway for sorting into lysosomes. And that is what Listerine is preventing. Listerine attaches a bunch of ubiquitin molecules into the ABCA1 protein such that it makes it, quote unquote, less visible to the HRS sorting pathway. And then the last two figures are about how all of this, everything that they've seen in vitro with cells in culture, they can see in vivo with animals, and how they, by manipulating Listerine and ABCA1 and all of that, they can actually aggravate or protect against atherosclerosis and heart disease and myocardial infarction. Yeah? So The last thought that I wanted to leave you with is that this paper was about a gene that is important in the context of Alzheimer's disease, but everything was done in cells that have nothing to do with Alzheimer's disease, and in a context, a biomedical research context that has to do more with heart disease than brain pathologies. But in the discussion, they have this paragraph in which they mention that it has been reported that mutations in listerine cause neurodegeneration in mice, and that enhancing ABCA1 recycling to the membrane could restore ABCA1 activity and enhance A beta degradation. So they just throw it out there that everything that they're showing that could help with cholesterol metabolism and cholesterol efflux from cells, and therefore diminishing the chance or the risk of atherosclerosis and heart disease, could be also applied to the brain, because the more ABCA1 that may be on the membrane of neurons and microglia could help with clearing A beta peptides and oligomers from the interstitial fluid. Yeah? So I went ahead and in our growing network, I added a node between listerine and ABCA1, basically saying that listerine can promote the levels of ABCA1. This is a great paper, super thorough in the science and everything. Unfortunately, it just explored this one connection, but it's a pretty firmly supported one. So, any questions, anyone? That's all I have for this first article. No? All right, so let's move on to the next. The next is titled Sleep Deprivation Exacerbates Microglial Reactivity and A beta deposition in a TREM2 dependent manner in mice. And in the introduction, what they tell us is that irregular sleep patterns have been associated to neurodegeneration, though it is not clear whether sleep changes contribute to the disease. I added that, i.e., whether sleep deprivation is a cause of Alzheimer's, or if sleep deprivation is simply an early symptom of a disease that is already taking place, i.e., there are consequences. Right? So you may appreciate now that many of these articles that we're discussing start with some sort of correlation, and the scientists exploring a little deeper to see if we can turn these correlations into causations. Okay? So irregular sleep has been associated, that's sort of a keyword that gives it all out. It's a correlation between sleep deprivation and early Alzheimer's. The question is, are sleep deprivation a cause of Alzheimer's, or is sleep deprivation a symptom of Alzheimer's, therefore a consequence? So studies have shown a correlation between sleep loss and increase in levels of A beta in the CSF, in the cerebral spinal fluid. So less sleep, more A beta in CSF, and we know now that that's bad. We also know that microglia are attracted to A beta plaques and can devour them. Right? So we've been talking all this time about how microglia will chew up these A beta plaques. And one thing that they introduce in the, a concept that they introduce in the paper is that there are variants of, and that's a mouthful, but TREM2, like, for example, the R47H variant that they use throughout the paper, that impair microglial chemotaxis towards plaques. The reason I mention this is because this becomes important later in the paper. Microglia, in order to devour these plaques, they need to go to them first. Right? So some previous work has shown that certain variants of TREM2 don't let microglia go towards the AB plaques. So we, as in they, asked, can sleep deprivation affect the A beta swallowing function of microglia? So can sleep deprivation make it harder for microglia to degrade AB plaques or beta plaques? And if so, is this effect of sleep on microglia TREM2 dependent? Or is it depending on very high levels of A beta? So right after reading the introduction, you realize that this paper is going to be a little bit of a nightmare. Because what these people are doing, unlike the previous one, is they're tackling two questions simultaneously. They're basically trying to connect sleep deprivation to lesser microglial A-beta clearing function, right? So that's fine. Establishing that connection should be okay. But then they're saying, and is this connection dependent on TREM2? Or is it dependent on the fact that there should be a lot of A-beta surrounding these microglial cells in the first place? Or both, right? So they're tackling two questions at once, and obviously that complicates the experiments a little bit. The reason why they're interested in whether or not any of this is affected by a lot of A-beta prior to sleep deprivation is because they can't shake off the back of their mind the possibility that sleep deprivation can only affect people who are already experiencing some of the issues related to Alzheimer's disease. In other words, that sleep deprivation affects very early stages of a disease that has been established by something else prior, right? And that's something that is always out there. This idea that there are certain environmental factors, like sleep deprivation, that can cause a lot of damage under certain conditions. And in this case, those certain conditions would be a prior deposition of large amounts of amyloid beta plaques, or at least the oligos in the brain. Okay? Any questions, anyone? All right. So, because they are going to intersect these two lines of research that have to do with having a lot more A-beta oligos in the brain with sleep deprivation, one of the first things that the authors wanted to make sure is that those two things don't intercept by themselves. In other words, they wanted to see, for instance, if you take a mouse. So we've talked about some mice models of Alzheimer's disease. We touched on one that overexpressed APP and presenilin 1, I think it was. So that's a model of Alzheimer's disease in mice. This is a different one. It's a 5 times FAD, life of me, I can't remember what that stands for, but it's one of the mouse models of Alzheimer's disease, meaning these mice naturally will deposit large amounts of A-beta oligos in the CSF, right? They do it because of their genetic background. And it is used as a mouse model of Alzheimer's disease because essentially what you're doing is you're accelerating a process that would otherwise take months and that may or may not happen in a wild-type mouse, right? So you're simply overriding the system and filling the brain with A-beta oligos. So the first question they ask is very simple. If I take these mice with a lot of A-beta oligos in their brain, do they show any sleep disorders, right? Because if that were the case, then there is no point in asking any more questions. These two things are intersecting by themselves, so there's no point in separating these two things, right? So what they see is that there is two comparisons that we need to make here. First, in both graphs, each graph measures something related to sleep that is a little different from each other. So the graph on the left measures the percent of time that the mice spent sleeping. And the graph on the right, as the label indicates, measures the sleep bout length. Mice will sleep in bouts. They don't have continuous night of sleep. They sleep and they wake up a little and maybe they move the whiskers. I don't know what they do. And then they go back to sleep and so forth. So these sleep bouts you can measure as well. Both are measurements of sleep quality, if you will. In each of these graphs, you'll notice that there is circles that indicate control animals, that you're letting them sleep by themselves, you're not doing anything to them, and then sleep deprived. That SD that we'll use throughout the slides stands for sleep deprived. So these are mice for whom I believe what the researchers do is they turn on the lights in the middle of the night. I should preface this by saying the following. With mice, it's the other way around. So mice are nocturnal. So they get active at night, right? And they sleep during the day. So one way of disrupting sleep is by either turning on the lights in the middle of the night so they are active, but now all of a sudden they're like, oh, I should go to sleep. And then you turn off the lights during the day, and they were asleep because that's what they do during the day. But then when in one of those waking ups in the middle of the night or in the middle of the day, they see that it's dark out there, and they're like, oh, I should go and forage for food or something. So that's how you mess up their sleep. That's how you cause sleep deprivation. Anyway, so first thing to notice is when you don't do anything to the animals, when you're not flicking lights on and off, they sleep about the same percent of the time, whether they're controls or they have a crap load of A-beta oligos in their brain. And when they do sleep, they sleep for the same bouts of time. Yeah? So that's one. The other one that they wanted to make sure is that with these flicking the lights on and off, that their sleep would be interrupted similarly. And in both cases, in the control background and in the A-beta super depositors, if you will, there is sleep interruption in both cases, right? And they measure that by seeing that the mice spend less of the time sleeping, and that when they do sleep, they sleep for shorter bouts. Okay? All in all, what this image says is we're going to be looking at A-beta deposition in the brain. We're going to be looking at the presence or absence of this TREM2, and our experimental control or our experimental variable will be sleep. But we just wanted to show you that there is no intersection between these things at the beginning, at least. Yeah? Cool. So then they actually go ahead and do the experiment. In the experiment, what they do is they take, here what you see is two graphs. They both reflect two aspects of the same thing, and that is microglial activation. So the graph on the left shows the percent of immunopositive. Sorry, these are, I'm so sorry, these are amyloid beta plaques. So this is plaque deposition in the brain. So there is a percent of immunopositive A-beta areas, meaning how much of an image is occupied by these A-beta plaques, and then the count of these plaques. These two go hand in hand, but the idea is with more A-beta deposition, you will have not only more plaques, so higher counts, but also they're going to be larger. So they're going to occupy a larger percentage of the area. So those are the y-axis in these two graphs. The x-axis get a little more complicated. So in the x-axis, you see three genotypic or genetic backgrounds. The first one is the mice that have the 5 times FAD, so they're producing a lot of A-beta oligos, but they're otherwise control for the TREM2. They didn't want to bother with TREM2, so they say T2 across the board, right? So they're wild type for T2. The other two mouse lines are the 5X FAD with that variant of TREM2 that we know is associated with poorer microglial function, and a straight out knockout. So a mouse that doesn't have TREM2, but that expresses a lot of A-beta. Both graphs show more or less the same, so I'm going to walk through the one on the left only. What they see is that in controls that sleep normally, there is certain amount of A-beta deposited. That's absolutely expected of these mice because these mice, by design, express a lot of the A-beta oligos, so plaques will form. But they also notice that when they sleep deprive these mice, there's a significant increase in both the size a number of plaques being produced in their brains. Right? So these mice overproduce A beta, and if they're sleep deprived, there is further increase in the number of plaques produced. What they see when they take out TREM2, either by having a mutant TREM2 that doesn't work or no TREM2 at all, what they see is that animals of those genetic backgrounds that can sleep normally, or that are allowed to sleep, I should say, show an increase in the number of plaques compared to TREM-containing controls, and that when you sleep deprive those, there is no further increase in either the size or the number of plaques being formed. So the baseline for plaque formation is higher in these TREM2 lacking backgrounds, and on top of that, when you sleep deprive those guys, there is no difference. Did that make sense? Yeah? In the legend to this figure, they say sleep deprivation exacerbates amyloid plaque deposition. So I can sort of agree with that by looking at this. Right? So sleep deprivation exacerbates. There is already amyloid deposition, but sleep deprivation will exacerbate it in a TREM2 dependent manner. Because when we take out TREM2, that exacerbation goes away. My question to you guys is, do you agree with this interpretation? So in a way, I know this is not exactly what you said, but I'm going to paraphrase. In a way, what you're saying is they did what we were suggesting before with the previous paper. You have an initial cause. In this case, it would be the amyloid plaque deposition caused by the 5 times FAD mutation. And we have an outcome, right, that is the formation of the plaques. And there is an intermediate that is TREM2, through which, I should say that the original cause is sleep deprivation, that through TREM2 is causing this exacerbation in plaque deposition. So we take out the intermediate TREM2, you don't see the effect anymore. Do we all agree? Take a minute to look at those graphs and tell me if you can think of an alternative explanation for what's happening here. It's good. This is not like a low-level concept, so it's fine. And I have a slide ready to address it slowly. The authors themselves alert us to an alternative possibility. So personally, I'm not quite sure what that cognitive disconnect there was between writing the figure legend and writing what they wrote in the results, because what they wrote in the figure legend is not all that airtight with the data. But what they wrote in the results is legit. So I'm paraphrasing here to summarize a little bit, but basically in the results they say the lack of sleep deprivation effect on A beta deposition in the knockout mice could potentially reflect a plateau on earlier saturation of amyloid deposition. Because remember, these guys lacked TREM2, and maybe when you take out TREM2 from a brain, one of the things that happens is there is a lot of A beta deposition, right? So, which is, in fact, we can see it. We see it here. When you take out TREM2, and even if those mice are sleeping normally, there is a lot more A beta plaque deposition. Right? So we take out TREM2, there is a lot more A beta deposition, and now we sleep deprive these animals, and there is no further A beta deposition, right? So what does that do to, in the context of the conversation that we were having before, the fact there is no further deposition kind of reinforces this idea that A beta deposition happens through TREM2 only, in a way, right? That TREM2 is the intermediate. We took it out and there is no further deposition. So what they alert us of here. So this idea that TREM2 is the mediator of this process is what fed the figure legend. They're concluding that TREM2 is the one responsible for this connection between sleep deprivation and A beta deposition. But in the results, they say, well, careful, because maybe there is a plateau of earlier saturation, and therefore, this earlier saturation might mask further effect of sleep deprivation in these mice. Basically, what they're telling us is maybe we don't see further A beta deposition because the system maxed out. Maybe the system can't take any more A beta plaques. Maybe we reached the maximum, right? So taking out TREM2 raises that floor, and then we add on top of that sleep deprivation. But if I raise the floor almost to the ceiling, I can't go any further than the ceiling. Did that make sense? Right? As we will see, they have additional experiments that kind of do support the model. So in the end, everybody's fine, everybody's good. But my question to you guys now is, how could we find out if we maxed out on A beta deposition? What could you do to these animals to find out if effectively we've reached the limit? What would you do?
How about if we increase the AB load?
Uh-huh.
If we increase it, like, for example, for like older mice, right? If we see no increase in the KO, like, we could see that.
Excellent. Excellent. So increase somehow the AB load or A beta load. And I was just about to say, yeah, but how would you do? And you said older mice.
Yeah.
That's it, right? Or what else could we do to increase the A beta load? Exact. ABC, for instance, or we could treat with drugs against ApoE. Exactly. Anything that we know that would normally increase the amounts of A beta deposition, we could try on top of these things. And the idea here experimentally is if you take a mouse that deposits large amounts of A beta to start with, you take out TREM2 and you either age it or give it ABCA1 inhibitors or ApoE inhibitors or some pro-neuroinflammatory treatment of some kind. And you see these now, a new cloud of data that are up here. Then that would be an indication that the system wasn't maxed out. And if you show me that, then I'm all in. I completely bought the argument that TREM2 is the one. Okay? What do we call these things that we do to show that our machines can still read outcomes or readouts? Yeah, what is it that we call these things that we do in our experiments to make sure that our equipment is still detecting changes? They have a name. Calibration would be for the low end, right? For whenever you want to see what the machine does when there is nothing to read. But then what do we call the things that we do so that we can make sure that the machine is still reading? Positive controls. It's the positive controls. So oftentimes when you see or when you hear a talk or something like that, you hear people asking, and what's your negative control? So that's a fairly common one. But a fairly common one as well is positive control. Whenever you make a claim about there was no further change or there was no augmentation or there was no signal beyond this point, one of the questions that you could ask is, well, how do you know that the machine can read beyond this point, right? Or how do you know that the brain could tolerate more A beta plaques? Maybe you've maxed out. So for that, we use these positive controls to show that we're fine, the system can still, there's room for more, right? All right, cool. So, let's turn on the page. I already told you that more data were coming that makes everything fine, and this is it. What they do now is they look at, before I misspoke, I said that they were looking at microglial activation. Here's where they look at microglial activation. So they use that IBA1 staining that detects microglia in the brain. And by measuring the percent of IBA1 area, they're basically measuring how many of the cells in a given region of the brain are microglia. So the higher the number, the more microglia infiltration there is. And they also measure, this is an important one, the percent of IBA1 staining, meaning presence of microglia, within the vicinity of A beta plaques. So this is measuring how close the microglia are getting to the A beta plaques. Looping back to this idea that microglia can engulf and degrade A beta plaques, but they need to get to them first, right? So quickly, what they show in these experiments, I'm gonna go through the image in the left because that shows something very similar to what's shown on the right. When you look at wild-type mice that don't deposit any extra amounts of A beta oligos in their brain, and you sleep deprive them, so this is the control mouse that sleeps normally, but then you sleep deprive them, there is a little bit more significantly, statistically significant increase in the number of microglia present in those brains. So there is microglia infiltration that is caused by sleep deprivation in control mice. But if those control mice didn't have TREM2, now those numbers stay flat. So here we're not raising the roof. We're not raising the floor, and yet the numbers are not going up. Does that make sense? In the previous experiment, we were raising the floor, so there was question of whether maybe we can go over the roof. But now, floor stays the same and the numbers don't change. You see the same thing here. So the reason why these yellow, orangey dots all go up is because now we're in the background of mice that do have that mutation that allows them to express a lot of A beta. So these mice naturally will undergo a lot of microglial infiltration. And even in those mice that undergo naturally a lot more microglia infiltration, sleep deprivation will significantly increase the infiltration by microglia. Right? But in those mice that don't have TREM2, no difference. Okay? The same profile applies to the microglia getting close to the A beta plaques. And therefore, the conclusion from these experiments is that sleep deprivation causes an increase in the number of microglia, and it makes them better A beta hunters, for lack of a better term, right? So these microglia in sleep-deprived animals are getting closer to, or more of them are getting close to the A beta plaques. So that's what sleep deprivation does to a mouse if they have TREM2. If they don't have TREM2, none of this happens. Okay? Cool. So then they need to address, the authors need to address what appears to be a little bit of a paradox. We're saying that sleep deprivation is making those brains have more microglia in them, and apparently they're better at tracking the A beta plaques down. They're getting closer to them, or more of them are getting closer to them. On the other hand, they're trying to push this idea that sleep deprivation will enhance the AD phenotypes. So these microglia there are more of them, and they're better at tracking the plaques down, but still we end up with more plaques and with more disease. How can we put the two together? I thought microglia could destroy these things. Now we have more of them, and they get closer to them, but nothing happens. So they decide to address what's happening to the plaques or the A beta oligomers once they're inside of the microglia. And what they do for that is an experiment very similar to the one that we discussed on Monday. They inject fluorescently labeled A beta 42, I think it is, into the brain of these mice. They wait a couple days, and then they use microscopy to see how much fluorescence there is inside of the microglia. Now here's where it gets a little counterintuitive. Turns out that when you take a control mouse here and you sleep deprive it, there is more fluorescence inside of the microglia. They call this co-localization of the A beta fluorescence with the IBA1 staining. So there is more fluorescence inside of the microglia. You're thinking, oh, these microglia are better at swallowing A beta. It's the opposite. What they claim is the more fluorescence you see inside of microglia, the worse those microglial cells are at degrading it. It's almost like they're incorporating that A beta into themselves, but they're not doing anything with it. Right? So sleep deprivation does that, and in these animals that naturally deposit more A beta, even though there is less A beta inside of the microglia, baseline, when they sleep deprive those animals, doesn't seem like much, but whew, it passed significance, so they can make the claim that it still happens in these mice with increased A beta levels. I'm going to point this out. I'm not going to give much away because I'm going to invite you to do one of those write-up or thinking at home, but I want you to start, greasing those wheels and think about why it is that in these mice that have a lot more A beta in their brains to begin with, there will be less fluorescence going into those microglia, okay? Because that may seem a little counterintuitive. But you don't have to answer now. We'll leave that for a homework assignment. Cool, so what do we have so far? We have that sleep deprivation is activating the microglia, more microglia in the brain, and they seem to be better at tracking down plaques, but at the same time, it's affecting their ability to digest these A beta oligos once they engulf them, right? So what the authors decide to do next is try and address what the genetic basis of all of these might be. And the approach they take is very common these days since sequencing technologies have become a lot cheaper and easier to do. They simply sequence the heck out of everything out of everybody. They took all 4 mouse lines. They let them sleep normally or sleep deprived them. And they extracted brains from each of these mice, groups of them at a time, and sequenced all of the RNAs that were present in them. So they're doing transcriptome analyses across all of the experimental backgrounds. Okay? And these things are very common to see in papers these days, these several ways Venn diagrams where you have each of the ovals represents the collection of genes that showed differential expression under a given experimental condition. So in this case, for instance, the yellow oval are the genes that were differentially expressed when we compare wild-type mice with a lot of A beta deposition in the brain and a wild-type TREM2. Right? So that was so we have the 5 times FAD and a wild-type TREM2. And what we're doing with these mice is let them sleep or sleep deprive them, compare the levels of RNA in their brains, and whatever genes show statistically significantly different levels of expression between these two groups, we call differentially expressed genes or DE genes, right? So all of the DE genes that we identify in that group go here. All of the DE genes that we identify for this group go here, and so forth. The reason we have these Venn diagrams that can get a little busy to interpret is because there can be some overlap. Some of the genes that are differentially expressed in these mice here, when you let them sleep or sleep deprive them, may be shared with differentially expressed genes that happen in a different mouse group, right? So these Venn diagrams are to depict precisely how many genes were exclusively differentially expressed in a group versus shared with other experimental groups. Okay? So far, so good? This one is going to get interesting. So we know that sleep deprivation causes differential expression in 537 genes. I'm telling you that. It's in the paper. It's a datum that I'm sharing with you. My question to you is, what do you think? When you add a lot of A beta, so the 537 are here in the blue oval, right? When you add a lot of A beta into a brain, will sleep deprivation change the expression of more or fewer genes? Do you think that we'll see change in expression of more than 537, fewer than 537? And in either case, do you think there's going to be a lot of overlap or they're going to be completely different? Seva says different because these mice here have a lot of A beta in their brain, and that will cause changes in expression of a different set of genes. Anyone agrees, disagrees?
I think if we add more, I think it's gonna add more genes.
More genes?
It's gonna cause a lot of like inflammation.
Uh-huh.
I don't
Maybe
know about the overlap.
you don't know about the overlap,
I
but
would say
let's go with that, the more genes. So Lara is saying, well, you're adding A beta, right? You're adding inflammation, so there's going to be more genes changing. So let's, for argument's sake, let's say that that this A beta trick changes the expression of 600 genes, right? So now the question is, these 600 and 537 are going to be completely different or mostly shared, and these guys just adding some. If I go with your model, for instance, I could say that of the 537 in the wild type control, I don't know, maybe 437 are shared. Only 100 are unique to this group, and then these here would be 163. Right? Because basically what you're saying is most of the genes are going to be the same. Adding all of that A beta is just adding more genes that are going to be outside of the blue circle, right? Or we could go, crazy, and we could say, no. It's actually 537 here. And then all 80 A beta is doing is adding 63, right? That's the extreme of your scenario, that you're adding something, so therefore you're going to be adding new genes that were not present in the original set. Ooh, excellent question. I don't know if we'll have time to go in detail, but there are statistical tests for basically asking the question, this degree of sharing, is it statistically significant or is something that you would find by chance, right? So there are mathematical tests for testing that. A case like this, I would say that since one set is completely contained within the other, that the overlap is complete, right? But the question is, what happens when you have a few out here, and some in the middle? Is that sharing just by chance, or is it significant? The mouse has so many genes, right? So if you change the expression of 2/3 of the genes in one mouse and 2/3 of the genes in the other mouse, there's going to be a lot of overlap, right? Did that make sense? All right, cool. So here are the results. The results are that actually most of the genes changed in the wild type. A few changed in the one with a lot of A beta. So that's 75 actually. So it's 537 versus 75. So a lot fewer genes changed in expression when you add a lot of A beta, and there were some shared. Whether that set of share is statistically significant or is something that you would have found by chance, we would have to run the test. We're not going to do that, and they didn't do it either. So what they conclude from this, or what we conclude from this, is that overloading the brains with A beta greatly dampens differential expression of genes. So before there were 537 genes going crazy, either up or down, but when you load the brain with A beta, that change seems dampened. And that's, by the way, going to be related to the question that I would like you to think about over the weekend. How come that we overload the brain with these things and everything seems a little more quieter? Okay? Yeah, all right. And partly shifts the effects of SD on gene expression, meaning that these 75 were not included in the original set. There were some that were a little different. So there's a slight shift, but it's not much. Then they asked the same question about whether brains have TREM2 or not. So we already said sleep deprivation causes changes in 537. What happens if I sleep deprive a T2KO, a knockout for TREM2? Will the genes that change in that case be more, fewer? And either case, will there be a lot of overlap or very little overlap? So what do you think? We have a mouse, we sleep deprive it, 537 genes change in expression. Now we do the same to a mouse that we took out apart. Okay. So the argument here is there will be more because you took out something, now the whole thing needs to adapt, so a lot of things are going to change. I like it. Does anyone would like to argue differently? I probably scare you by saying I like it, but the argument in the opposite direction is, well, you just took out a part, so it's almost like, it's a crude analogy, but you take one wheel out of a car, it can still kind of go. It's going to sound ugly and you'll probably be pulled over, but it can kind of go, right? So that's the argument for saying, I expect that a few additional genes change, and the ones that change are going to be mostly contained within this, right? Crucial for me to, exactly. So it can go a number of ways. Which way did it go? Let's find out. So they saw that a lot of genes change in expression. So now we're talking 563 total compared to the 537. So as many as in the wild-type background, but they were completely different. Only 70 of those genes overlap between the two sets, right? So taking out TREM2 completely changed the transcriptional response of the brains to sleep deprivation. Josh, in a way, you're arguing, well, you took out TREM2, so the whole thing needs to readapt. It's no surprise that now when you hit it with sleep deprivation, a new set of genes will change, will go up or down, because a new set of genes is being accessed. Maybe. But the observation is that. What I wanted to do with this is not so much about you figuring out what genes change in a sleep-deprived versus not, depending on wild-type background or beta deposition, TREM2, and so forth, but mostly to get a sense for what it is to engage with these Venn diagrams, right? Because they're all over the literature. This is the same for the sleep deprivation of the A-beta depositors with or without TREM2. And again, the effect on gene expression was dampened and zero overlap. So it's almost like he completely changed the mouse by taking out TREM2. Anyway, what I want to say is that I want you guys to get comfortable with this because these types of figures are more and more common in papers these days. And if you continue on a path that has to do with understanding modern genetics, you're going to come across these things all the time. So the way to read these things is to try and understand what each of these intersections mean, and from there to try and get a sense for what genes you should pursue further. And I wanted to do that exercise with you. So the authors found rather surprising that the transcriptional changes differ so much across backgrounds. In other words, so little overlap. There's those 70 there and a few genes in the 2 ways intersect, but nothing in the 3 ways or the 4-way intersect, nothing. So these genes that are changing, going up and down, are not shared among conditions. The whole thing is changing. I have a question for you so that you practice this whole engaging with these Venn diagrams. We know sleep deprivation causes microglial activation and therefore inflammation, we need to assume, regardless of whether a brain has a lot of AB floating around or not. But this is only if the mice have TREM2. This brings us back to these figures in which both the wild-type mouse, the blue circles and little diamonds, go up when you sleep deprive them, and the A beta overexpressors, the yellow and orange circles and diamonds, go up when you sleep deprive them. But in both cases, if you take out TREM2, everything stays flat. Right? So that's what feeds that statement on the top. In this map, where might you look for the genes that are responsible for this microglial activation if you know that sleep deprivation triggers this microglial activation in mice that have TREM2, and regardless of whether or not they deposit a lot of AB? Where would you look? Which of these groups of genes would you say, I'm going to look into this. I'm going to list them out, and one by one, I'm going to start doing experiments. Jacob. The 70 down here? Any takers for the 70? Why? It is independent of AB. It's true, because neither the, so we're talking about the both wild types, so no AB there. Cool. And they both what?
They both have
Maybe this, this thing is, it doesn't show really good, but this says T2KO.
Oh, okay.
Yeah, but you're on the right track though. You're on the right track, so sure. But if they remove TREM2, then because they show up in the TREM2 knockout set, they cannot be responsible for this activation. The 11. What are you going to say, 11? Say 11. Huh?
The yellow.
The yellow or the 11? The 11. The 11. I had a slide for that. The 11. Why? Because the 11 are genes that are changed regardless of the presence or absence of the A beta. This is lots of A beta. This is no extra A beta, right? These genes here could be those genes that happen because of the extra A beta. But the prompt says this microglial activation happens whether you have a lot of A beta or not. This down here is the opposite. These down here happen only when you don't have a lot of A beta. So it's the 11 that you look at, because those are the ones that happen regardless of A beta levels, but only when you have TREM2. Does that make sense? Cool. All right, guys.